fermentation, even when there's been the forming, that's going to be the tissue that will give rise to 2 or 4 weeks. And have that ability to have unspecialized cells that are divided and divided to give rise to more tissue that then will have specific things.
Yes. A question about the cascade in ferns and how the calcium channels open. Where does the calcium concentration?
Oh, okay. So you're talking about germination in a fern spore, yes? So when light is absorbed by that fern spore, red light is absorbed by, what's the name of the red light receptor? Right. So when light is received by, absorbed by the phytochrome, that causes a conformational change, and it then is able to trigger this whole biochemical cascade that we then see ultimately as, oh, that spore germinated. Right? And part of that cascade is the opening of calcium channels. So that calcium is going to come from the outside world. There's calcium in the water, in the forest, in rainwater, etc. Right? So it's going to come from outside. Yes?
What causes sporangium to mature? What causes sporangium to mature?
What causes a sporangium to mature? Are you talking about in the fern? Okay. So in a fern, you saw the underside of the fern leaves. You saw those little sporangia, brown, dust, whatever, when it was in stripes or blobs or balls or whatever, all the different morphologies. Each sporangium has a life cycle, all right, and it has to go through both the surface cells, the sporangia themselves have to mature. Inside each sporangium, the spore mother cell is going to undergo meiosis and give rise to spores. All of that depends on both environmental triggers and genetic triggers. So different species of ferns are reproductive at different times of the year. This sort of goes beyond what we said in class. We may have talked a little bit about this in lab. But we don't usually see sporangia maturing in the ferns in the woods now. The leaves have died back, etc., and they're not producing sporangia. So plants can be very seasonal. And so their environmental triggers, are there enough light, is the temperature right, etc. And so as that sporophyte matures, that fern that's growing up, it will then have the right temperature, have grown large enough to then be able to undergo that part of the life cycle. And for each sporangium, it's going to be based on that species of plant and the environment.
I'm sorry, go ahead. Can you explain why, when you have the monosporic and the tetrasporic development, why the central cell becomes triploid for the monosporic development, and for the tetrasporic development, the central cell becomes pentaploid?
That happens when? When can we look at a mature embryo cell? Okay. So we've got right? We've got our embryo sac. I'm not telling you now whether this, I'm not going to differentiate immediately whether this is in a monosporic plant or in a tetrasporic plant. But here we are in our embryo sac. All right, let's make this a microbial side, right? All right, so if I say that this is a monosporic, mature megagametophyte, right, here we are. We've got the ploidy of every nucleus I've drawn in here, and a monosporic one is what?
Haploid.
Haploid, right? So I've got N, haploid, blah. What's going to turn, so right now, what's the ploidy of this central cell? N plus N, all right, you've all got it. When and only when does this then become a triploid nucleus? After fertilization, all right? So pollen tube grows down, pollen is landed way up on the flower stigma is like the roof of class at this mag or something. Pollen lands on there, the right species, it's triggered, absorbs water, blah, 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 grows the pollen tube, comes down, And that pollen tube grows then through a synergid, right? And it's got the two pollen sperm nuclei in there, right? One of those is going to end up in here, fertilizing this, and one is going to be here, fertilizing here. That's when the three nuclei will fuse.
Okay.
A haploid polar nucleus that originally came from this end, a haploid polar nucleus that originally came from this end, and a haploid sperm nucleus that's delivered by the pollen tube to give us, I don't know, draw it over here, to give us inside. Now we have one triploid nucleus after fertilization. Yes?
Just a little clarification question. So from the pollen tube, there's supposed to be a tube cell and two sperm.
Yes. So
And
the tube nucleus is somewhere here.
So
that degenerates.
So it degenerates?
Mm-hmm. The
Okay.
tube nucleus has done its job. It has directed the tube growth through the micropyle, and it will then just dissolve, degenerate. Does not fuse with anything, does not transcribe any genes, does not do anything.
Same thing with the vacuole.
Yes,
it all disintegrates.
yeah, and the whole pollen tube will just disintegrate, will dissolve, will disappear. It has done its job of getting the two sperm nuclei where they have to go.
Thank you.
All right, so now if we switch gears and say this is a tetrasporic embryo sac, what are the ploidies of these two nuclei before forget fertilization yet? What's the ploidy of these two nuclei? Right, so I'm going to make one of them bigger, right? So we have a 3N and an N. So how and when does this become pentaploid?
Right.
Only after fertilization, when one of those sperm nuclei has landed on the stigma of this tetrasporically developing plant, grown down, chemotaxis, etc., gets down here, comes in, and now we've got a haploid sperm nucleus. They all come together and magic, whatever, star, and we've got in here ovule. So the central cell's nuclear ploidy is going to depend on before or after fertilization event. The same way that the egg is haploid until and unless a sperm nucleus gets delivered to it, in which case then it's the zygote that's diploid. And that's the first diploid cell of the new sporophyte generation. And that pentaploid, if we're a tetraspore, or triploid, if we're a monosporic nucleus, is the first nucleus of what? What does that central cell give rise to? Endosperm, right? So you can have either triploid endosperm or pentaploid endosperm, depending on what kind of plant this is. Has that plant followed the evolutionary path where its embryo sac uses all four of the spores, tetrasporic development, or is that a plant that has allowed three of those spores to die, and one spore, that monospore, is following the monosporic development? And that's what I mean. There's no way just to look and just say, oh, this one is monosporic, this one is tetrasporic. You'd have to be able to look and count chromosomes, you'd have to know about the plant. I will never expect you to know. I will always tell you this is monosporic if I'm asking you a question about it, or this is tetrasporic. Or I will say, draw me a mature embryo sac for a tetrasporic plant. Include ploidy. Right? Now fertilize it. Include ploidy, or what would change, or something like that. That I will always ask you. So again, I'm begging you, read the questions really carefully. And I will tend to, I always do, highlight or underline or bold or italicize tetrasporic, or underline, before fertilization. So read it out loud in your head, not out loud. Read it in your head with the emphasis where I'm putting it. So you go, wait, why is this word in bold? Tetrasporic. Oh, okay. She really wants me to know that this is tetrasporic and that the question is about tetrasporic. What does that mean? You're going to have to pull that part out from the story, from the study. And therefore, I know that I'm going to have to be talking about something. Tetrasporic, there's going to be triploid nuclei there, and At some point, I might get pentaploid, endosperm nucleus, etc. Okay? Question?
So on the exam, if you say before fertilization, that means that there's only going to be 2 1N nuclei.
N plus M in the central cell. Right. So I could very well ask you to draw an embryo sac. Or I'll give you an embryo sac and say label it, or something like that. And in that case, think of all the different ways that you could draw it. I might just say, draw me any embryo sac and make it correct and tell me what you're drawing and why, but I tend not to do that. You know, I would tend to give you, say, draw a tetrasporic embryo sac before fertilization. And then you should realize that I want you to do, it's got, the antipodals are there, the synergids are there, the egg is there, the central cell is there, right? If I'm asking you to draw me, what did I just say? Tetrasporic before fertilization, all right, and show ploidy. If I say ploidy, you have to put nuclei in, right? And if I ask for ploidy, you can either do it by writing N if this is, oh, I said tetrasporic. 3N, you know, you can write the number in, you can do it with arrows. These are 3N antipodals. Just make it clear, you know, that you know what the cells are, and you know what the nuclei are, and what the food is. That clear? Yes.
After fertilization,
Yes. So after fertilization, the synergids would disintegrate ultimately, and the antipodals would disintegrate ultimately. They don't do it in milliseconds. It's not like the sperm come in and, but they will die. They will not continue to be. You will not find them soon after that. Yes. What do the antipodals actually do? That's a good question. What do antipodals actually do? They don't really have a function in terms of, they don't become endosperm, they don't have a fate in the future of this developing sporophyte plant, they don't contribute to the embryo itself, they don't grow and turn into a leaf. You know. So what's the point of having antipodals? It's something that you guys ask a lot in different classes. What's the point of that? If an embryo, especially in development, if an embryo develops something that then dies, why on earth did it ever develop in the first place? Wouldn't it be easier just not to have antipodals?
Do they provide nutrients?
Maybe, and that would be a good, she said, do they provide nutrients? Perhaps the dissolving antipodals can help add some nutrients, or as they may be synthesizing some material, but they're not known as real nutritive tissue. Exactly. You've got it, when you have mitosis, when you have meiosis, there's certain kinds of nuclear divisions that happen. And you're going to get daughter nuclei, and or meiosis is going to give you 4 nuclei, etc. And so you can end up with nuclei in the cells that may not have a fate later on, but were necessary for the process to evolve. So if I said, what is the future fate of antipodals, you would write, they don't contribute to the development. They don't become part of the embryo, they don't become part of the endosperm, their future fate is to die. Okay. But a better, a longer term, if we were having a really evolutionary discussion or a philosophical discussion or talking about how and where the plants, how did all of these things arise, we could talk about how you might have gotten to have these divisions to allow for one nucleus to migrate from that end, had to make some triploid nuclei in order for one to get to the center, etc. And maybe in a former iteration, maybe, a million years ago, those antipodals did something. And then the embryo sac didn't need it, and so that. Yes.
You said that the zygote releases an inhibitory signal that prevents the other sporophytes from developing.
In which, I want you to be sure so that not everybody doesn't get confused. In what organism are you talking about? Great. Okay, just because we're here deep in angiosperms,
right.
So I want to just make sure that everybody knows. She's now switching us back to ferns, and her question was something about, or she
I was
said,
asking whether it has a name, like.
oh, the actual chemical.
Yeah.
No, I never gave you any of the actual components, don't know. So, but the fact is that a zygote on a fern gametophyte, the first egg that gets fertilized in an archegonium, even if there are 5, 10, 20 archegonia on that particular bisexual gametophyte, the one that gets fertilized first is going to release an inhibitory chemical that inhibits any other embryo from developing. And that's the way that a tiny heart-shaped gametophyte ensures that it will be able to provide the nutrition necessary for the growth of one and only one sporophyte that grows from that fertilization.
I have a question back to the angiosperms with the pollen tube. I think in the notes said the tip of the tube there's a lot there's a calcium influx which triggers exocytosis at the tip of the vesicles. I was wondering if you could
Exocytosis of vesicles at the tip.
okay.
Right. So vesicles, exocytosis always implies, the word exocytosis means a membrane-bound vesicle fuses with the plasma membrane of that cell and releases its content to the outside. That's what exocytosis means. It's often triggered by a rise in intracellular free calcium. When you think about what it does, that exocytosis event could be incredibly important for what it spits out to the outside world, that is for the contents of that vesicle, or it could be incredibly important because by exocytosing, what does it give to that part of the cell? It gives membrane, which increases the surface area, which is just what you want to do if you have a tip that has to grow. So the exocytosis at the tip of the pollen tube of those vesicles is providing membrane that allows it to go from here to here to here to here. It's giving you more surface area at the front.
So more on this topic. So when you have a mature pollen grain, could you explain a little bit about how the process and so essentially like you have like the two sperm cells and then you have the tube nucleus. So what exactly is like where is the growth coming from exactly? Like the
Okay, so let me erase these angiosperms, these embryo sacs. So let's say we have a pollen grain that has landed, right? And it has been triggered to take in water and gets the signals from on the stigma, etc. So here's our stigma, right? And it now starts to germinate. It goes through that, there's water uptake, there's all the signals that are necessary. It can then grow through and break open at weak spots in the pollen grain wall. There are going to be little weak spots. And so what we've got is the growth. If we look inside here, here is the cell plasma membrane, and then we start to get tube growth. Okay? Breaking through a weak spot in the spore pollen and pollen grain wall. In this one cell, right, we have sperm, and we make them funny shapes because they are funny shapes, right? And we have a tube nucleus, which tends to lead the way. The tube nucleus tends to be near the front of where that pollen tube growth occurs. And so if we watch, what's happening here is it can take calcium up from the stigma tissue. Right? That calcium causes exocytosis of a gazillion, I can't draw them tiny enough, little vesicles. There's cytoskeletal elements that help direct them towards the front. And we're going to get this tube getting longer and longer and longer and longer. What's happening is these three nuclei, the tube nucleus and the tube sperm, are going to be staying near the front. So I have to kind of draw it as a time lapse. I can't just leave these three things here, because they don't stay there. So if we look next, all right, and we've got the pollen green wall stays there, right? That's not gonna go down through the stigma, through the style tissue, etc. We're going to see that now the tube is longer, and we've got our tube nucleus here, and we've got our sperm nuclei there, and exocytosis is happening at that front. You saw the movie where there was very clear cytoplasm at the front, and that's where exocytosis is happening. If we then watch later when the tube is this long, right, we've got our tube nucleus, we've got our sperm nuclei, and we've got more exocytosis happening here. And now we've got this big vacuole in here, because the cell just can't make more cytoplasm. It can take in calcium, it can take in water, and it can fill this with water to sort of allow for most of the cytoplasm to be at the front of this growing tube. And this tube you saw in pictures and movies and animations has a huge job to grow down. In your dissections, think what was the distance from the stigma to the ovary in your lily when you broke it open? You dissected your flowers, right? It was roughly this tall. Think about one pollen grain having to grow that far down. That's a huge amount for it to grow. And a lot of that volume of the growing pollen tube is water. Right? And the important stuff is just at the very tip.
So what's the tissue then that's being pushed by the pollen tube? Yeah, what is it? Is it the...
You mean what, in the stigma and the style, what it's growing through?
No, from the mature pollen grain, that
right,
part
that's the one, this is one cell that's growing big and big and big.
So it's just one cell that's...
Yes, well, it's three. I mean, you have the two sperm, but then you have the one, this is one cell. That's what I'm trying to draw. There's the plasma membrane, it has the two sperm nuclei in there. All right, they're either nuclei or cells, depending on how much membrane they have around them. We didn't spend a lot of time, and as I keep saying, it depends on the species of plant. But we've got two sperm nuclei and a tube nucleus in one cell. And that one cell is changing its shape. It's changing its shape in an incredibly dramatic way. So here's the round spherical pollen grain, and the cell is, the size of a softball, if there's magnification, etc. And what we're going to watch over time and what happens is that one round cell changes its shape by sending out a protrusion that gets bigger and bigger and bigger. It's not making more cells. It's not building up any other tissue. That cell is changing its shape, extending out this extension called the pollen tube that is lipid bilayer plasma membrane. That's what I'm touching. If I touch it, that's what's pushing through the transmitting tissue of the style. That's what's sniffing those lure proteins that are coming from the submergence, so it knows to grow this way and not that way or that way. Right? That's what's allowing this one cell to do this absolutely unbelievable task. Right? It really is extraordinary that a pollen grain can make it, that the one cell can grow directionally and tremendously to deliver its contents. The tube nucleus will die, but those two sperm nuclei have to be delivered to that embryo sac we just erased, so that one will fertilize the egg and give rise to the zygote, and one will fertilize the central cell and give rise to the endosperm. Some plants have really short, stubby distances between the stigma and the ovary, and the pollen grain doesn't have to grow an incredibly long tube. You watch tubes in lab, right? Didn't you? Did you all get to see that? Yeah? How? Wasn't that impressive? You didn't see cell walls forming. You didn't see that it's making a big tissue. You saw that it was sending out a long protrusion, and in some cases it might whirl around. I mean, it's on a microscope slide. It's not getting the right signals, etc. All right? But in the stigma of the appropriate species, it would have grown straight down till it gets to the entry of an ovule by that microscope. Yeah.
We saw in the video that every time the pollen tube knew which place to go, so we didn't discuss it. Like he knows that the chemicals are coming from this region, but we don't know that. How does those kids supposed to?
Oh, yeah, so they made it seem in that movie, in the animation, it's really cute and it's funny, and I can't remember what weird music they had for it, but it's as if each one, you know, I'm gonna get that one, I'm gonna get that one, I'm gonna get that one. If you look at actual photographs of pollen tubes, excuse me, if you make them fluorescent and you make a slice through at some point, after the many pollen have germinated on that stigma and grown down, in a species of plant that has many ovules in its ovary, right? You'll see that they cross over each other, two maybe trying to get in, you know, it's not that neat and clean. It's not like this one comes down, you know, I mean, this one goes, I'm going to number 1, I'm going to number 2, I'm going to number 3, number 4. They're all, there's a lot of chance in it, right? And there also is, they're getting many signals, and so this one holding in on this particular micropyle will then block that micropyle. Micropiles aren't very big in most species, and so a pollen tube going through it can sort of keep more of that signal from emanating out, and so it doesn't necessarily plug it up, but it can sort of block the signal that's coming from that particular synergid in that particular embryo sac, so that then another pollen tube that's coming down minutes later, hour later, whatever, isn't going to smell as strong a signal, and it will then keep going to the other. Right? But there's a lot of chance, and sometimes ovules just don't get fertilized. Pollen may not land on it. Only a few pollen of the right species may land on it. That transmitting tissue might have plunked out for some reason and might not provide enough nutrients, enough calcium, enough whatever, and that fruit will never form. Those ovules will never be fertilized, and that is it for that flower. It will not go on in the seed. Right? But overall, evolutionarily, they do it all the time, and there's a lot of success. Yes, ma'am.
We wanted to know like what causes the anther to dry up and split, and how do they know when the pollen is ready? Like,
How does the anther know when the pollen is ready?
yeah, like because you know when they split the pollen, they just
Right. So again, that's part of the overall, that's sort of the bigger picture that we didn't spend a lot of time talking about. What are the environmental signals? And a lot of this has to do with specific plant hormones that we're going to be meeting soon. Right? So in some ways, you've got a lot of the anatomy and the physiology without necessarily getting the cause yet. And I hate saying that. Oh, wait a few more weeks. Or we'll get to the genetic control later or something. But that is one of the things where both environmental signals and hormonal signals within the plant are what's going to allow the maturation of any one anther on any one flower on any one species, etc. And so there are hormones coursing through the tissues of the anther. They're environmental, you know, it has to be dry enough or warm enough or sunny enough or whatever for that plant to then flower. You know, there are going to be all kinds of environmental yes.
Oh, I have a question, like going back to the fern.
Okay.
When we're talking about the trigger for two-dimensional growth, I think like the trigger was blue light.
Mm-hmm.
But I was wondering if there's any chronology behind it, like in terms of like red light and blue light.
Right, well you have to have red light. So a fern, we're back in fern world. Right? Fern spores will land on the dirt, in the soil, on the auger, in your plate, whatever, right, into a moist environment with the right kind of temperature, etc. That fern spore must experience light of varying qualities at different times in order for that spore to give rise to a normal, happy, healthy, successful gametophyte. So the first quality of light that must be there in order for it to germinate is red light that is received and perceived by phytochrome. That allows for germination. And there's that biochemical cascade, et cetera, we talked about. And so you first get a one-dimensional sort of filament growth. 5, 6, 7, 8 cells, depends on the species of fern, et cetera. But you know, you get a little file of cells. That's all that will continue growing. That gametophyte will never mature, will never make antheridia, will never make archegonia, if it stays only in red light. But if it can perceive blue light, once it has generated a filament that is 4, 5, 6, 7 cells, depending on the species, if that tip cell then absorbs blue light, that's going to trigger it to then change the orientation of the mitotic spindle so that now that cleavage burrow is gonna cut the other way, and we're gonna start getting two-dimensional growth. And from then on, it can do 2D growth. So the chronology is, it must have red light in order to germinate, and it must need blue light then for 2D growth. So you can provide a fern spore out in nature is getting often both blue and red light, and it will use that red light first, and then it will have cells that are able to then use blue light because they have generated the blue light photoreceptor called cryptochrome, right? And they then can receive a blue light signal and respond to it in a really interesting way of transitioning to two-dimensional growth, changing the orientation of that signal. So she's asking, is the word prothallus and gametophyte interchangeable? Prothallus tends to be used once we're starting to make two-dimensional growth, and prothallus is often used when it's multiple cells, but I use it interchangeably. Okay. Yes.
Are there male amoristic gametophyte? I mean, female amoristic gametophyte, but no.
Oh, good question. No, in ferns, they make either bisexual gametophyte or male only. Yes.
And does the antheric have a part to play in the production of only male gametophyte in amoristic?
So antheridium is what causes a fern spore to develop into a male-only amoristic gametophyte. So every fern spore of the species we're talking about, right, I'm putting in a little caveat because there are some real oddball things out there. But everything we're talking about, that every fern spore that lands has the potential to give rise to a bisexual gametophyte. And that's the normal, expected, future fate of a fern spore, is to give rise to a bisexual gametophyte that will make both antheridia and archegonia. However, that normal fate can be biased, can be changed, can be controlled by this extracellular environmental hormone, antheridium, that can overwhelm the normal growth pattern of a spore that is germinating into a gametophyte and cause it to develop into a male-only, antheridia-only gametophyte. Yes.
So if it's extracellular and environmental hormone, where is it coming from?
Well, I mean, it's released from, where is it coming from? I think its action is coming from the outside. Where does it come from? Who or what? Right, the meristic gametophyte releases antheridium. And then it gets out into the environment. It's outside, and a spore then landing in water, in an environment that is rich in antheridium, right? Its future fate is transformed by having that hormone around. And it will only develop into that little strap-shaped one that only has the antheridium. Question?
Is antheridium the same chemical that signals the other archegonium to not fertilize? Or is
No. Antheridium is not the same as what maintains only one sporophyte developing per gametophyte. It's a different chemical, a different inhibitory chemical. Okay. Question? Yes?
When the two sperm are formed, do we have the cell membrane around them?
So her question is, when the sperm form in a pollen grain, in a mature pollen grain, are they sperm nuclei or are they sperm cells? That is, do they have a cell lipid bilayer plasma membrane around them and a cell wall, because we're talking in the plant world? And here's where I've tried not to be too fussy because it varies in the different kinds of angiosperms. All right? Some of them do make sperm cells, when they divide, when that generative cell divides into, it gives rise to 2 sperm cells. But in some species of angiosperm, that cell wall doesn't really form, and that plasma membrane may or may not be there. All right? And so I tend to talk more about the importance of the sperm nuclei, or just say sperm without saying whether it's sperm cell or sperm nuclei. And so I will never ask you that specifically, because it's, in many angiosperms, yes, it's got cell membrane around it and cell wall. So we call that mature microgametophyte of 3 cells, microgametophyte, right? It's got the tube cell and within it 2 sperm cells. But in fact, in some species, they are more like nuclei than they are like fully formed cells. Yes?
Could you please explain about tetrad of spore and fern? Tetrad of spore
A tetrad of spores where?
in fern.
In the fern. All right, so what is a tetrad of spores in fern development? First off, where are we going to find spores being formed in a fern? In the sporangium. All right, a single sporangium will have within it diploid cells called what that are going to give rise to spores? Right, they're going to be spore mother cells. So a spore mother cell is a diploid cell that undergoes what process to give rise to 4 cells? Right, it's going to undergo meiosis. Now, we went through meiosis in class on Friday, but I'm not going to ask you the details of meiosis. You don't have to know leptines, IPs for this test, but you really need to know when and where meiosis occurs, because we've been talking about it for the entire plant lectures, and the fact that meiosis gives rise when a cell nucleus undergoes meiosis, the result is 4 haploid nuclei. That's the result. Meiosis is the nuclear division that gives rise to haploid nuclei. So in ferns, in those beautiful brown crusty things on the undersurface of the fronds, sporangia, there are cells that when they undergo meiosis give rise to 4 haploid spores. We call that a tetrad, right, because they're 4 of them together. And I actually hate the word tetrad because it's used in so many different places where something gives rise to 4, or there are 4 of something. So you'll hear tetrad when we talk about chromosomes, when we talk about spores, when we talk about whatever. But it is good because it lets you know there are 4 of them. So meiosis in ferns, in a spore mother cell, undergoes meiosis to give rise to 4 haploid spores that together form a tetrad. Does that answer your question? So, tetrad of microspores is a product of meiosis of a spore mother cell in a sporangium on the underside of the frond. And each one of those spores then will undergo a maturation process to get its own little spore pollen wall put on it, etc. There's a little tri-leaf scar showing where it was stuck to its other 3 sisters, and then go forth in the world when that annulus dries up and it flings it out to form a gametophyte that's either male only if it's under the influence of anthragogen, or bisexual if it's not. Oh, we talked about how those fern spores can separate, didn't we? Mm-hmm. Mm-hmm. So there's enzymes that can help separate them. Right. Yes.
Is the sporopollen then what creates the ridges on the spores that we looked at, or?
Yes, so those ridges, so sporopollen is a waxy particular polymer substance that's very hard, can be waterproof, etc. And sporopollen can be made made in ferns, it can be made in angiosperms, and it can surround spores or pollen, hence the name sporopollen. And that's what is sculpted into those different shapes. And if you look, I showed some SEMs of different pollen. We looked at fern spores and saw ridges and try these scars. That sculpting, that shape, those ridges and valleys and pores and blobs and pimples and whatever are all sporopollen. That's the outermost cell wall material that's going to be on the outside of pollen or on the outside of spores.
So suspensors inside of come from the basal cells, or they come from the
Okay, so when we have a zygote, she's asking about where the suspensor and basal cell comes from. Right? So when an egg gets fertilized into a zygote, right, that then undergoes its first division is what kind of division? Unequal, meaning you get one big cell and one small cell, right? And that small cell goes on to give rise to the embryo proper, and the big cell gives rise to the basal cell and the suspensor. So both the basal cell, which is all the suspensor cells, and all of the zygote cells, embryo cells, come from that first initial diploid zygote that starts dividing in the embryo sac after fertilization.
So I don't know what my question is, but can I draw it?
Sure.
Okay, so in a spore, this is the spore pollen, this is the triloid scar. What are these things?
Sporopollen.
So that's spore pollen and this is spore pollen?
Yeah. The whole thing. So say that we're gonna take a tennis ball and that is the spore cell itself. Okay, that's got tennis ball has the furry part of the tennis ball. I'm trying to think ahead. Is this a good analogy? All right, good. You got a tennis ball or something, right? That is lipid bilayer, and inside it is nucleus. It's a spore. We're talking about firm spore right now, right? So it's got a haploid nucleus. It's got cytoplasm in there. There is the cell. Now take some clay dip, which is sporopollen, and within the sporangium, it gets layered and it gets stuff put on top of it. That sporopollen is going to make a thick coat that will protect each one of those spores. And sometimes it has ridges, so make some snakes out of your Play-Doh and put it on and make it look like, etc. And that's what the sporopollen is. It's part of the cell wall that gets thicker and more crinkly and more ridgy and more convoluted, whatever word you want to use. Look at pictures of fern spores. Just Google it. Google image search. Fern spores, SEM, scanning electron micrograph. You'll get beautiful ones, or just regular photographs. And you'll see that some of them have the species you looked at. You saw that there were things like this here, whatever. All of that surface, anything I could, my finger were tiny, anything I could rub my finger on and touch on the outside, that's sporopollen. That's the outermost layer of that spore. Right? Those ridges, those bumps. Other species may have blobs on it as opposed to lines or whatever. So look at the picture. But that morphology is based on the species, and that's one of the ways that good fern biologists would be able to tell you, oh, if you give them 3 different samples of spores or a mix, those that have the ridges like that, that's Ceratophrys. These are, that's the only fern genus I know. These are other ferns. These are other ferns, okay? And they would be able to sort them. In the same way that in the angiosperm world, way over here, if you ask an angiosperm pollen biologist, they would be able to look at that mixed, beautiful picture we saw of different species on a stigma and say, oh, tulip, beech tree, or magnolia pollen, based on its shape and how the spore pollen was laid down. Yeah.
Could you explain what the megasporangium is?
What is the megasporangium? All right, so sporangium in general. What is a sporangium? Just in general, what's a sporangium? A place where spores are going to form, right? Okay. And in a fern, we only have one kind of spore. So the sporangium has that funny, shape, whatever, it flings out, whatever. That's where spores are made by meiosis. But in angiosperms, we're making 2 different kinds of spores one kind of spore is going to ultimately be able to give rise to a gametophyte that gives rise to an egg. And the other kind of spore is going to give rise to a gametophyte that's going to give rise to sperm. So in angiosperms, the fact that there are these two different kinds of spores that form, we use the prefix mega if it's ultimately heading female, and micro if it's heading male. And they are sporangia in different parts of the flower. So in the anther, there are sporangia within which spore mother cells undergo meiosis to give rise to spores. What do we call that? What prefix would I put on it if it's in the anther? What kind of sporangia is it? It's a microsporangium within which microspore mother cell undergoes meiosis to give rise to four microspores, which then go through microgametogenesis to give rise to a pollen grain. In another part of the flower, where would this be? In the ovary. In an ovule, there is a cell called a megaspore mother cell inside a megasporangium that undergoes meiosis to give rise to megaspores.
So essentially what you're saying is like you have the ovule, and then within the ovule you have megaspore. Is that like a coating, or is it just megasporangium is like the home for the megaspore?
The megasporangium is an actual structure. Right?
Okay, so
it has the megasporangium is ovary. This is a megasporangium, and inside it is a megaspore mother cell.
so it's a layer of cells.
This whole thing is the megasporangium.
Oh, okay.
Just like if we go back to fern world, that little brown thing that has a cute annulus, that's the sporangium. It has jacket cells, it has lip cells, right? It has nutritive tissue in there, and then it has spore mother cells. This is the megasporangium, and it has, each megasporangium only has one megaspore mother cell inside it. They can undergo megasporogenesis, give rise to how many megaspores? 4, by the process of meiosis, right? So you've always got to just think about ferns and angiosperms really do the same kinds of things. I mean, overall, all plants have this alternation of generations. They have diploid sporophyte tissue, they have haploid gametophyte tissue. Gametes are haploid, spores are haploid, sporophyte tissue is diploid, etc. Right? And in ferns, the reason we start with ferns is because there's only one kind of spore, and because you can see the gametophyte with your naked eye, well, if you have good eyes, and certainly under the dissecting scope, where you can see those little tiny green things on the ocra, and you can see the big fern, and you can see both parts, the alternation of generation. The sporophyte has cells that undergo meiosis to give rise to spores, that give rise to gametophytes that make gametes, the archegonium and antheridium, that can give rise to a zygote, the next part of the sporophyte generation. Zygote is the first cell of the new sporophyte generation. When we go to angiosperms, we've got to now realize, we've got two different pathways, and both anatomically and physiologically and just location-wise. It's happening in two different places. They're both happening in the flower, but one is happening in the male part of the flower, and that can be microgametogenesis, microsporogenesis. We're making microgametophyte, right? And in the female part of the flower, we've got mega. What's going on? Okay. But overall, sporogenesis is always going to be the process that gives rise to spores, and in flowers and in plants, that's always going to be meiosis gives rise to spores. In plants, meiosis gives rise to spores. Meiosis gives rise to spores in plants. Make it a t-shirt, or something. And that's By meiosis, haploid cells are formed that are spores, and those spores then give rise later on to gametes. And that's different from how animals do it. Alright? Yes?
So basically megasporangium plus the integument is equal to organ.
Yes, exactly right. So here, if we put now the integuments on here, and this little stalk that attaches it to the ovary wall, this is now an ovule. Exactly right. And this whole thing in here, this whole thing is what's going to mature into a seed. So a seed is going to have whatever happens inside the old megasporangium cell wall, which is going to be the embryo and the endosperm, cotyledons, etc. Ultimately, it's going to be the megasporangium wall, and the integuments are going to harden into the seed coat, or form into the seed coat, whatever, whether it's fleshy or hard or crunchy or think about all the different seed coats that you have. Yeah, the last one.
Oh. Did anyone else have like a question?
Go for it.
For the fate map in the fourth cell station experiment, you mentioned one of the cells can be to like foot. So could you like
So the foot is what is going to anchor that developing sporophyte into that gametophyte. It basically holds it and anchors it and can absorb nutrients from that gametophyte itself. So the heart-shaped gametophyte, one of the main reasons only one sporophyte can develop from any one heart-shaped gametophyte is that that gametophyte is passing necessary vital nutrients to this developing sporophyte. And it does that through the foot. The foot anchors it, the foot can absorb nutrients, sugars, etc. from the gametophyte tissue. It's going to stay there. It ultimately won't be part of the adult fern, but it helps anchor it.
Yeah, because I was wondering what it looked like.
Oh. Let me get my, make sure I make sure my picture looks good. So you've got it in your textbook, you've got the archegonium, all right, is going to have in it the zygote, right? I'm not going to draw, all this. And the zygote gives rise to the 4 cells, once this has been fertilized, etc. And one of these becomes the foot. But you have to remember where we are. We're in the gametophyte tissue hanging down. So these are green cells, green photosynthetic cells, 2 cells thick or 1 cell thick, depending on the species of fern and how and where. And close to the archegonium, they tend to be a little bit thicker. So this foot is going to develop into, and sort of the whole archegonium gets distorted by this growth, and it ultimately ruptures, and the old archegonia cells will disappear. But this foot will end up being kind of like this, anchoring it within this tissue. Right? We have a root that comes down, we have a shoot that comes up around, etc. All right? And so you're going to be able to see that this helps anchor it in and absorb nutrients from the rest of the food. All right. So the exam's Tuesday. You still got time to study. I'll try and answer email questions, but you also should have good notes, all of that. Read the questions